who is Ghulam Muhammad. Iqra Ghulam Muhammad. You can present your presentation. Oh, I'm sorry. I'm just announced as Iqra. Can you present your presentation?
Assalamu alaikum everyone. My name is Iqra Ghulam and I am from Department of Zoology, Rifa International University. Today I will present my research work entitled Effect of Green and Chemical Synthesized Cerium Oxide Nanoparticles on Blood Profile and Liver Enzyme of Serenius marigala.
Ikra,
This
your
study
screen is
focused
not
on
completely visible. I think you are on mobile. Is this fine if I play your slides here? Yeah, now it's good now. It's not good
yeah,
now. It's good now. Please start again.
okay. Now we start?
Yeah,
Okay. Assalamu alaikum everyone. My name is Iqra Ghulam and I am from the Department of Zoology, Rifa International University Faisalabad. Today I will present my research work entitled Effect of Green and Chemical Synthesized Cerium Oxide Nanoparticles on Blood Profile and Liver Enzyme of Serenius marigala. This study focuses on understanding how emerging nanoparticles affect aquatic organisms and environmental health. Water pollution. Water pollution is one of the most serious environmental challenges worldwide. Rapid industrialization, excessive agricultural practices, and urban waste discharge continuously introduce contaminants into aquatic systems. These pollutants disturb water quality, affect aquatic biodiversity, and ultimately pose risk to human health through consumption of contaminated water and fishes. Therefore, monitoring aquatic pollution is essential for the environmental sustainability. Traditionally, water pollution studies focus on heavy metals and pesticides. However, in recent years, emerging pollutants such as pharmaceuticals, microplastics, and nanoparticles have gained attention. Nanoparticles are particularly concerning because of their extremely small size, high effective and ability to penetrate biological membranes. Cerium oxide nanoparticles are widely used in catalyst, fuel additions, medical applications, and drug delivery systems. They possess unique redox properties, allowing them to switch between oxidation states. Well, beneficial in industrial and medicine, this same property can cause oxidative stress when released into aquatic environment. The toxicity of nanoparticles strongly depends upon how they are synthesized. Chemicals synthesized involve toxic solvents and reducing agents, which may remain attached to the nanoparticle surface. Green synthesized use plant extract or biological agents make it more environmentally friendly. However, green particles are more automatically safe and must be scientifically evaluated. Although many studies have examined nanoparticles' toxicity, very few have compared green and chemical synthesized cerium oxide particles in freshwater fishes. This lack of comparative data represents a significant research gap, which this study aims to address. Here are some resources of nanoparticles. Serenius marigala is an important freshwater carp species widely used in aquaculture. It is nutritionally valuable and economically significant. Because fish are in constant contact with water, they respond quickly to environmental changes, making them ideal bioindicators of aquatic pollution. function of Serenius marigal. 16 healthy fishes were selected and accumulated before examination. The exposure period of 28 days when chosen to observe subcritical toxic effect under the controlled laboratory conditions. Fish were divided into 5 groups, 1 control group and 4 treatment groups, exposed to 2 concentration of green and chemical cerium oxide nanoparticles. After 28 days, fish was examined. Blood samples were collected from hematological analysis, while liver tissues were preserved for biochemical and histological experiments. Blood parameters are sensitive indicators of experimental stress and change. Red blood cells, anemia, and white blood cell alternative reflect immune response. A significant decrease in red blood cells, hemoglobin, a significant decrease in red blood cell hemoglobin observed, particularly at low concentration, indicating stress-induced anemia. Increased white blood cells indicated activation of the immune system in response to the nanoparticle exposure. Evaluate liver enzymes indicate liver cell damage and metabolic disturbance, especially in chemical synthesized nanoparticle exposure. They observed toxicity may be due to react of the oxygen species generation led to oxidative stress and cellular damage. Chaotic nanoparticle exposure may reduce fish health, affect aquatic productivity, and pose food safety risks. In conclusion, cerium oxide nanoparticle cause dose-dependent hematological and liver toxicity, with chemical synthesis show greater harmful effect. So, the chemical synthesized cerium oxide affect more toxically to the fish as compared to green cerium oxide. In conclusion, cerium oxide nanoparticle dose are more safer biological synthesis than green cerium oxide. Thank you.
Hello, Iqra.
Yes, ma'am.
I have a question. Are there differences in blood profile between green and chemical synthesized nanoparticles?
Sorry, I cannot understand your question.
Are there differences in blood profile between green and chemically synthesized nanoparticles?
Yes, ma'am. A huge difference between concentration of red blood cells and white blood cells between the green and chemical cerium oxides जो groups हैं
Okay, thank you so much, Ikram.